microorganisms that are living in the various natural habitats, they are constantly being exposed to fluctuations in nutrients. Through the evolutionary processes, all these organisms, they have developed certain mechanisms by which they can compensate for such environmental fluctuations. Most of the bacteria, they alter their gene activity. This alteration of gene activity is actually responsible for optimization of growth and reproduction. So whatever may be the habitat in which these microorganisms are living, these microorganisms can change their gene activity in order to carry out an optimum level of growth as well as reproduction. This can be carried out with the help of two types of genes. First set of genes is called as regulated gene. As the name indicates, these genes, their activity is controlled in response to the needs of a cell or an organism. So as the habitat of that particular microorganism changes, organism can regulate the expression of these set of genes. Such type of genes are referred as regulated genes. The second type of genes are called as constitutive gene. These constitutive genes, these are also referred by another term, housekeeping genes. Whatever may be the environmental conditions, in all environment, in all situations, these constitutive genes or housekeeping genes, they are always expressed. They are never shut off. For example, protein synthesis and glucose metabolism. These genes, these are constantly being expressed. Expressed means here we are talking about transcription followed by translation. So out of the all genes that are present in the bacteria, some are always expressed, hence called as constitutive. Others, their activity is controlled in response to the needs of that particular organism. If in a particular environment the bacteria requires to uptake a particular nutrient, then the genes that are required for uptake and metabolism of that nutrient, they are expressed. Such type of genes are called as regulated genes. In order to adapt themselves to the changing environment, bacteria, they turn on and off the genes within them. If particular bacterial cell turn off the genes, this is done usually in a situation where the products of these genes are not required by the bacteria. So whenever a particular product or a group of products is not required by the bacterial cell for its growth, for its metabolic activities, then it turns off those genes. Whenever a product is needed, then a particular gene or a set of genes is turned on. So bacteria generally have an on-off mechanism for regulation of genes within them. This turning on or off of genes, it requires an interaction between regulatory proteins and the DNA sequences. Before discussing various important details of regulation of gene expression with respect to some important operon systems, let us try to understand what is inducible gene expression. Whenever a particular bacterial gene it is turned on by addition of a substance, by addition of a particular substance, then such type of gene is called as inducible gene. The regulatory substance which is controlling the expression of this particular inducible gene is called as an inducer. Inducers, they actually belong to a class of molecules which are called as effectors. So ultimately that means inducer, it is a type of an effector molecule, it is a regulator molecule. Its presence is responsible for turning a particular gene on. That means a particular gene will undergo the transcription in presence of inducer. Such type of genes are called as inducible gene. Now these inducers that are controlling the expression of this inducible gene binds to the DNA of that particular bacterial cell at a particular position. This particular position is called as the controlling site. So inducer will bind to this controlling site that is present on the DNA of that particular organism. Thereby, it allows the expression of this gene within that particular DNA molecule. So such type of genes are called as inducible genes. So whenever the inducer is present, presence of inducer and binding of that inducer at this particular site, controlling site on the DNA molecule, will allow that particular gene to undergo transcription, and transcription will be followed by translation, resulting in formation of that particular protein product. So we can summarize that inducible genes, these are expressed. Now here the term expressed means both transcription, which is followed by translation. This particular type of gene expression, inducible gene expression, is possible only in absence of a repressor or presence of an effector or inducer molecule. So in this case, if we discuss the situation, here we don't have the repressor. Here an inducer is there which is responsible for induction, binding at this site and then allowing the transcription and translation to occur. Okay. Let us try to understand the induction phenomena with respect to the lac operon that is present inside E. coli. E. coli has the ability to grow in a very simple medium that is consisting of salts, nitrogenous source, and glucose. Glucose is a source of carbon and energy in this cultivation medium. If instead of glucose, any other sugar is present, for example lactose or some other sugar, then the other enzymes are synthesized that are helping in utilization of lactose, or instead of lactose any other sugar is present. That sugar, it can be utilized with the help of enzymes that are rapidly synthesized by the bacterial cell. So depending upon the type of the sugar, whether it is glucose, lactose or some other sugar, enzymes are rapidly synthesized inside the cell for utilization of that type of sugar. For example, if the cultivation medium contains only lactose, in order to utilize lactose as a source of carbon and energy, the lactose that is present inside the cultivation medium needs to be taken up by the bacterial cell. In order to utilize that lactose, three different enzymes are required. First is beta-galactosidase, second is lac permease or lactose permease, and the third, that is transacetylase. The function of this transacetylase enzyme in metabolism of lactose is not completely understood. So here we can understand the lac operon enzyme activity with the help of this particular image. So a bacterial cell, if it is present in a growth medium with lactose as the only source of carbon, in order to utilize this carbon source to carry out its growth, this lactose needs to be taken up from outside of the bacterial cell to the inside. When it's taking up this lactose from outside to the inside, it requires help of certain proteins that are present within the plasma membrane. So plasma membrane will have the permease proteins within it. These permease proteins are responsible for transport of lactose from outside of the cell to the inside of the cell. So lactose is brought inside the cell with the help of permease proteins. Once it is brought to the inside, then this lactose, since it is a disaccharide, it cannot directly enter into the glycolytic pathway. It needs to be split into monosaccharides. Lactose is a disaccharide made up of two components, glucose and galactose. So this glucose and galactose that are linked together by the glycosidic bond, this glycosidic bond between them should be disrupted. Disruption of this bond is brought about by the enzyme that is called as beta-galactosidase. So beta-galactosidase will split this glycosidic bond present in between the glucose and galactose. Thereby, it splits the two sugars, glucose and galactose. Glucose directly enters into the glycolysis. You know glycolysis. The step starts with glucose. Glucose converted to glucose 6-phosphate, followed by many other intermediates, ultimately to pyruvate. So glucose, it is directly entering into the glycolytic pathway or glycolysis, whereas the galactose, it is converted to glucose first. After conversion to glucose, then it enters into the glycolytic pathway. The lactoperon genes, they are able to split the lactose in presence of lactose. This lactose, even though it appears to be an inducer of lactoperon system, resulting in production of beta-galactosidase and lac permease, the actual inducer is different. The lactose, as soon as it enters into the bacterial cell, some of the beta-galactosidase converts a small amount of glucose into allolactose. Allolactose then induces the lactoperon gene, resulting in production of beta-galactosidase inside the bacterial cell. So, even though the apparent inducer, it appears that presence of lactose is responsible for induction of lactoperon genes, resulting in synthesis of beta-galactosidase and other enzymes. It is not the actual inducer. The actual inducer is allolactose. And how this allolactose is synthesized? It is synthesized from the lactose. Lactose is acted on by beta-galactosidase, is converted to allolactose, and then this allolactose induces the formation of beta-galactosidase from lactoperon genes. So all the lactose that is present inside the cell may not be utilized for splitting of lactose into glucose and galactose. Some of the lactose is utilized for conversion to allolactose, which is the actual inducer, whereas lactose is an apparent inducer. Jacob and Monod, they were the first to propose the operon model for regulation of genes within the bacteria. The example that they discussed was of lactoperon. Operon is nothing but a cluster of functionally coordinated genes. Expression of the genes within an operon is regulated with the help of operator and regulator protein interactions. The operator region itself as well as the promoter. The lactoperon of E. coli can be regulated in two ways: by negative regulation with the help of repressor molecule, positive regulation with the help of catabolite activator protein cyclic AMP complex. This operon is said to be inducible control mechanism. In the present slide, we are going to discuss how this lactoperon, it is controlled with the help of repressor. Within the lactoperon system, there is a Lac-I gene. This is a separate operon system. Lac-I gene has its own promoter. This promoter is very weak. Because of that reason, when an RNA polymerase comes and binds to this weak promoter of Lac-I gene undergoes the transcription, but that transcription occurs at a low level. Hence, very few molecules of Lac repressor protein, these are formed from this Lac-I gene after transcription and translation. So within a cell, this Lac-I gene is responsible for synthesis of few repressor molecules. So product of Lac-I gene is a repressor molecule which is always present in low amount inside the cell. The reason behind its low number is a weak promoter. RNA polymerase, when it is binding to this promoter, it carries out the transcription at a very low level, resulting in formation of few repressor molecules. Now here it is important to note that LAC-I gene, it is a constitutive gene. That means even though the lactose is present or absent, in both the situations, the repressor protein is going to be synthesized. The LAC-I repressor protein is followed on the other section of a DNA molecule where the lac operon genes are there. Lac operon genes, they have a promoter, operator, and followed by three structural genes Z, Y, and A. LAC-Z, LAC-Y, and LAC-A are the structural genes. They are responsible for synthesis of LAC-Z synthesizes beta-galactosidase, LAC-Y synthesizes lactose permease, LAC-A synthesizes transacetylase. These enzymes, they are required for uptake of lactose from outside of the bacterial cell to the inside. Once it is taken inside, its processing is done with the help of beta-galactosidase enzyme of LAC-Z and then utilized as a carbon and energy source by the bacteria. The three important genes within the lac operon systems, these are clustered on the lac operon genome section. They undergo the transcription to give rise to a single mRNA. Now the situation here is three genes are present inside that lac operon system. Three genes means here three structural genes in the form of a cluster. This cluster will undergo transcription. Their transcription is going to form single mRNA. This single mRNA has an information for all the three genes together. Hence it is called as polycistronic mRNA or polygenic mRNA. So here we have the mRNA that has been formed due to transcription of lac operon genes. Lac operon genes has three genes, LAC-Z, LAC-Y, LAC-A. Three structural genes are present within the lac operon. After the transcription of the Z, Y, and A genes within lac operon, the mRNA that is synthesized will have information for LAC-Z protein, LAC-Y protein, and LAC-A protein. So ribosomes will bind to the 5' end of this polycistronic lac mRNA, will carry out the translation. So this section is going to result in production of beta-galactosidase. So when ribosomes, they load onto this mRNA molecule and start the translation, the first enzyme that will be produced is beta-galactosidase. The second section is going to lead to production of permease. So these ribosomes can slide along the mRNA molecule from one section to the another. This section is going to result in formation of lac permease, which is involved in the transport of lactose from outside to the inside of the cell. The third section is responsible for transacetylase enzyme. The function of this enzyme with respect to lac operon system is not completely understood. So these two components, beta-galactosidase and permease along with transacetylase, they are translated. These proteins are produced from the polycystonic lac mRNA, which has been produced from lac operon genes Z, Y, and A. The first situation that we are going to discuss in the lac operon is the absence of lactose. When lactose is absent inside the cultivation medium, in that situation, as we have already discussed, the LAC-I gene, since it is constitutively expressed, as soon as the transcription of this LAC-I gene is going to occur, lac mRNA will be produced. Again, this is not a lac mRNA, this is lac repressor mRNA. So LAC-I gene will only undergo transcription, will produce lac repressor mRNA. Lac repressor mRNA, by binding to ribosomes, will undergo the translation to generate the lac repressor proteins. This lac repressor protein has an affinity for this operator. If the repressor blocks this operator, then the RNA polymerase cannot bind to the promoter and then progress through the structural genes. So no transcription of Z, Y and A section of operon is possible when the repressor protein is binding to the operator. Does that mean the synthesis of all these three components is completely shut off? No. These genes are being expressed, but their expression, it is at a very low level. So even though here we find that structural genes are not expressed, operon systems in case of bacteria, they are not completely shut off. At the basal level, at a very low level, these genes are still being expressed. So the repressor that is bound at this particular position to the operator, it binds to this section. When it is binding, it will not allow the expression, but sometimes it falls off from that operator. That means it unbinds from this section. So when it falls off from this section, it allows some time for the RNA polymerase to carry out the transcription of these three genes. Transcription of these three genes followed by translation will produce the beta-galactosidase, lac permease and transacetylase. So repressor, its binding and unbinding from this section allows for synthesis of a very low amount of these three enzymes inside the bacterial cell. The second situation is the presence of lactose. When lactose is present, then in that situation, the repressor protein that is synthesized by the lac-I gene will combine with the allolactose that is produced from the lactose. Now, allolactose is the actual inducer. It is produced from lactose by beta-galactosidase. So, low amount of beta-galactosidase which is present inside the cell, it converts the small amount of lactose to allolactose, and that allolactose will combine with the repressor molecule. On combination of this inducer, that is allolactose, with the repressor molecules, the repressor will get inactivated. Its affinity for the operator is reduced. When its affinity for the operator is reduced, it will not bind to the operator. If a repressor is not binding to the operator, operator is free, then in that situation the RNA polymerase, it can carry out, it will bind at this position at the promoter, and when it passes through this section, it will transcribe like Z, Y and A region, resulting in formation of lac mRNA. This lac mRNA has information for lac Z gene, lac Y gene and lac A gene. When ribosomes will come and bind to this 5' end of lac mRNA, they will result in formation of beta-galactosidase enzyme from this section, permease from the middle section, that is lac Y section, and transacetylase from the lac A section. So we can say that in presence of lactose, the beta-galactosidase enzyme, low level of beta-galactosidase enzyme that is present inside the cell, converts the lactose into allolactose. Allolactose then interacts with the repressor, inactivates it, so that now the operator is free. Repressor cannot bind to this operator. In that situation, the RNA polymerase will bind to the promoter and then carry out the transcription of lac mRNA. On translation, it is going to give rise to beta-galactosidase, permease and transacetylase. When only lactose is present, in that situation, positive regulation of lac operon can also be carried out. This positive regulation can be carried out with the help of a cap protein which is called as catabolite activator protein. This protein is actually inactive. It is converted into an active form by combination with another component which is called as cyclic AMP. Cap protein on combination with cyclic AMP forms an active complex. This active complex can bind to the cap site which is located within the promoter. Binding of this complex at the cap site within the promoter, it actually helps in binding of RNA polymerase to the promoter. So RNA polymerase, its binding is facilitated by presence of cap cyclic AMP complex at this cap site. So polymerase will come and bind, will carry out the transcription of this lag Z, Y and A section, which is followed by translation with the help of ribosomes, which lead to formation of beta-galactosidase, permease and transacetylase enzyme. So function of this catabolite activator protein is that it helps in ensuring that operon, it is on, its transcription is on when lactose is only the source of carbon. But if glucose is present in the same medium, so there are now two sugars, lactose and glucose. In that situation, the cap protein is not activated. This is not able to remain in active form because the cyclic AMP concentration falls down in presence of glucose. This is an image of the operon system which shows us the cap binding site which is located within the promoter. So here you can see I gene is present, followed by cap site. This is actually located within the promoter. Here we have the RNA polymerase binding site. When cyclic AMP and cap protein complex is present at this particular cap site, then the RNA polymerase can come and bind to this position. Then we'll carry out the transcription of Z, Y and A genes. When the cultivation medium has both glucose and lactose, in that situation, organism, that is E. coli, it prefers the utilization of glucose since this particular glucose can directly enter into the glycolysis. In case of lactose, it needs to initially synthesize permease, beta-galactosidase, which will convert these ultimately to glucose, and then it can enter into the glycolysis. Whereas if it gets directly the glucose, then it prefers the utilization of glucose since it can directly enter into the glycolytic pathway as compared to the lactose. Certain initial steps are required for the breakdown of uptake and breakdown of lactose, then only it can enter into the glycolytic pathway. This is not the case with glucose. So if there are two sources of carbon inside the cultivation medium, that is glucose and lactose, E. coli prefers to utilize glucose over lactose. When glucose, it is diminished, in that situation, it will start production of enzymes that will utilize the lactose. So when glucose is present in the cultivation medium along with lactose, the operon genes, they are repressed, they are not allowed to undergo the transcription. This is taking place because of one important mechanism in which the glucose, a readily available carbon source, it inactivates the enzyme that is called as adenylate cyclase. Now this adenylate cyclase, it is an enzyme which is converting ATP, adenosine triphosphate. This is adenosine triphosphate. This adenosine triphosphate, when acted on by adenylate cyclase, it is converted into cyclic AMP. So if glucose is present, this enzyme is inactivated, adenylate cyclase. And if adenylate cyclase is inactivated, will there be enough concentration of cyclic AMP? No. So when both glucose and lactose are present, in that situation, glucose is inactivating this particular enzyme, so low level of cyclic AMP will be there inside the cell. Or cyclic AMP might be absent inside that bacterial cell. In absence of cyclic AMP, there will be no activation of catabolite activator protein. Inactive catabolite activator protein cannot bind to the operator. If it is not able to bind to the operator, then RNA polymerase binding is not facilitated. So no transcription of Z, Y and A genes within the lac operon is possible when the cyclic AMP is absent. Cyclic AMP will be absent since glucose is inactivating the adenylate cyclase. So in presence of glucose, the concentration of positive regulator, that is cap cyclic AMP complex, that is able to bind to the lac operon for increased gene activity is reduced. So once the ATP is converted to cyclic AMP, this cyclic AMP is again converted by phosphodiesterase enzyme to 5' AMP. So glucose removes remaining cyclic AMP. So whatever is initially present, it will not allow the conversion of ATP to cyclic AMP. That's okay. But small amount of cyclic AMP may still be present inside the cell. So glucose removes remaining cyclic AMP by activating an enzyme which is called as phosphodiesterase. Phosphodiesterase enzyme will convert that cyclic AMP to 5' adenosine monophosphate. In this manner, when glucose and lactose both are present, in that situation, the enzymes of lac operon, they are not synthesized. This is possible because of one reason, that the concentration of the positive regulator is cap protein along with cyclic AMP. This is not allowed to form. In absence of that, increased gene activity of lac operon is not possible. Presence of glucose reduces the amount of cyclic AMP. Cyclic AMP amount, when it is reduced, then cap cyclic AMP complex will not form, no binding to the cap site on the operon, no increased expression of lac operon gene. At basal level, still the expression is going on, but increased transcription of Z, Y and A is not possible when both carbon sources are available, glucose and lactose. When the glucose gets depleted, when the glucose amount goes on reducing, then there should be the synthesis of lac operon genes. So when glucose will be reduced, adenylate cyclase enzyme, its inhibition, its inactivation will be lifted. Now active adenylate cyclase enzyme will be available. This adenylate cyclase will then convert ATP to cyclic AMP. Cyclic AMP that is produced will then combine with the cap protein, and this particular cap protein along with cyclic AMP will bind to the cap site, facilitate the binding of RNA polymerase enzyme to the cap site, to the operator, ultimately allowing the transcription of Z, Y and A genes, leading to production of beta-galactosidase, lac permease and transacetylase enzyme. So in this manner, the lac operon can also be positively controlled with the help of cap protein along with cyclic AMP.